Yeah, so the story I'm going to tell you today is basically it shows you the pleasure and productivity of following up a natural history question. That's, I guess, like Bruce, it's been the story of my life. I watch creatures and think about, try to figure out what they're doing, and then ask questions about how they do it, and maybe even why they do it. Why is harder, of course. But so today I'm going to tell you about harvester ants, which is a large genus of New World ants. They occur from southern Canada all the way down to Patagonia. In the United States, there's about 25 different species, including one here in Florida, Paganomyrmex badius. It's a funny name because Paganomyrmex means bearded ant and badius means beard. So it's a bearded ant. And like most of the genus Paganomyrmex, they collect seeds and they store them in underground chambers. And the amount of seeds that constitutes their diet is quite variable. But in the local Florida species, badius, they do store, it is a very major part of the diet, especially with seasonal variation. So let me give you a brief introduction. The habitat is primarily the sandhills of Florida, all the way up to, I think, South Carolina. Oh, wait a minute. I brought my own pointer. As you know, oops, that's Bruce's ant vial. What did I do with my pointer? There it is. I have deep pockets, so there it is. Put Bruce's ants back in my pocket. Ants in the pants, right? Okay, so the habitat is obviously on the Apalachicola National Forest, and where's my little dot? Oh, there it is. Not too strong, is it? This is an area of the ANF which I call ant heaven. And it's a very good habitat because, well, I think I got gypped by this pointer. Yeah, here we go. There's a creek here, Fisher Creek, which is about 30 or 40 feet lower than the flatwoods over here. So the water table is depressed from going here to there. In the flatwoods, it's about a meter below the ground surface. At Fisher Creek, it's obviously the water table. And in between, it can be up to 15 feet deep. And so that makes that site very dry. And it originally, at least, supported a very prairie-like ecosystem with Nolina, yucca and prickly pear and all those prairie dry adapted plants. And one of the creatures that loves this kind of habitat is the fire ant. The harvester ant. Got to keep my ants straight. And what you see here is a map of about, I don't know if you can see the little dots, the pointed dots. That is those are the GPS coordinates of about 400 colonies that I tracked for 10 years. So I know a lot about their life cycles and their life history and so on. So, and you all know, of course, that it's a fire climax kind of forest, and it depends on regular fires for its persistence. This is the ant, and the ant has two body forms. One are the ordinary workers are these what we call minim workers. They're, actually it's a big ant compared to other ants. And then they have these very large major workers whose function has been speculated to be opening seeds and that kind of stuff. And the peculiar habit that really sets this ant apart is that they go out into the habitat and they collect little bits of charcoal, which they bring back and decorate the nest with. And I know you're going to ask me, why do they do that? Right? Why? Thank you, David. Why do they do that? I have no idea. But I've had, I had a student test 8 different hypotheses, none of which were supported. And there was a 9th one, which one of my students and I tried to support, but we never got the right circumstances. So the short answer is, I don't know. The reason they collect seeds is because, especially at some times of the year, they husk these seeds and they chew them up into ant bread. And if you crush a seed, you know, it starts the amylase hydrolysis and it makes it more palatable. That's how we make bread. So they chew up the seeds and they feed them to their larvae. Okay, so that's why they collect seeds. This is a chamber deep in the nest, and this chamber happens to be full of sexual ants. These are the males. That's a sexual female with wings, but there's also larvae and pupae and so on. So it's a classic ant life cycle. They collect a lot of seeds. These are the seeds from a single chamber of a harvester ant colony. And a colony can collect up to 300,000 seeds. That's like a kilogram. And it's a lot of seeds. So this, of course, is the link between the ant and the native plants, why I'm here, right? Because they do collect a lot of seeds. So it's probably a fairly large effect on the success of plants. The nest that they dig is typically very deep. So they're dependent on these deep soils with a very deep water table. And you guys know what turkey oak is, right? In the sandhills, if you want to find harvester ants, look for the turkey oak. If they're there, they will be associated with turkey oak because that's also has the same hydrological requirement that harvester ants do. In the flatwoods, you occasionally find them, but you know, then the nest is only that deep, and every couple of months they get flooded out. So anyway, this is a plaster cast of a large harvester ant colony. This cast actually sits over in Bio 1, Bio 1 on the 2nd floor across from the elevator, which right now it's under renovation, so you can't see it. But it's an unusually big colony, right? But here are the statistics about it. It was built by about 5,000 workers in about 5 to 6 days. And in doing so, they moved 20 kilograms of sand. And the total weight of the ants is about 20 grams, so you can figure out through the math. There are 135 chambers, and the sum total of these vertical tunnels is about 10 meters. So it's a big job, and they do all this in 5 or 6 days, about on average once a year. They move the colony, they make a new colony about once a year, and they do the whole thing, move the seeds, the brood, even the charcoal in 5 or 6 days. It's actually kind of amazing. I think so anyway. So I've had several partners in crime here. This is my former student, Christina Kwapich. And that is me in the bottom of a hole, which is where I've often found myself. And Christina did a lot of work on several different aspects of harvester ant biology, some really beautiful work. Then these are 2 assistants that I hired. One is Nicholas Hanley, who I put in charge of culturing the seeds for germination and so on and so forth. And then Julio Dominguez, who did a lot of the IDs using the FSU herbarium in part, but also other sources. So he was important in identifying all the seeds. So we published a paper with pictures of all the seeds that the ants had collected from 2 different dates. One is a study I did in 1989, where I excavated 31, I think, colonies, complete colonies of harvester ants and collected all their seeds. And the second one from 2014 when, you know, again, we collected a lot of seeds. So, I mean, you guys are the botanists, so you can look at some of these names. And anyway, if you want the complete list, it's published in a paper that you can find on my website as a PDF. You can find all my papers as PDFs on the website. So those of you who saw my exhibit in the Junior Museum, no, wait a minute, Tallahassee Museum, this was a display of the seeds from 11 different harvester ant colonies. Each jar contains all the seeds from a single colony. That's a lot of seeds. And each seed is a minimum of one trip, right? Minimum. And of course, a lot of the workers also come back empty-jawed. So we've compiled a list of all the species. This shows only the 21 most common ones. You guys are experts here. You can probably know what the common name of these things are, but in 1989, the most common species were, let's see, that's down to about 2%, so 8 or 10 species. In 2014, the frequencies differed, and we can speculate on why. It's probably because the site is undergoing succession, and it's a lot more, it's shadier, and a lot of the ground species have changed in frequency. So that wouldn't be too surprising. There's also another 38 species not listed here that are less than 0.1% in frequency. So, okay, you guys are botanists. You know that when you dampen a seed, it germinates, right? They sprout. That's how you get them to start. So the answer, piling all these seeds into damp chambers in the ground, and it is damp. So, you know, my question was, how do they keep the seeds from germinating, right? So that's how I started this little quest. The question was, how do the ants keep the seeds from germinating? So Christina and I did a simple experiment in which we put seeds taken from harvester ant colonies, and we put them in these petri dishes with a porous one, so that if we put that dish in the ground, it would equilibrate in moistness with the surrounding soil. And we buried them at 4 different depths. Here's the little stacks, and left them for December till April. And when we excavated again, sure enough, there were quite a few that germinated. So the question was, how do the ants keep them from doing that? Well, I thought about it a while. And you know, sometimes you can have these barriers to thinking, right? I was asking the wrong question. So the question is here's these giant seeds. I mean, look, that's a major worker. It's a lot bigger than the ants. How do they're supposed to eat these seeds. How do they do that? Well, it's like you eating an elephant. You got to get through the skin, right? So, whoops, wrong pointer. Eventually, thinking about it, I thought, I'm asking it the wrong way. It's not how do they keep them from germinating. They're waiting for them to germinate because these seeds are so big that they can't get through the seed coat. So they're depending, this is a hypothesis, right? They're depending on the seed to germinate before they can eat it. That sounded a lot better. So in fact, the evidence was there all the time. It's another one of those examples where you're kind of blind, you know? You see it, you don't understand what it means. In this case, if you look at the seeds from the chambers, look how big they are. I mean, they're huge ones. If you look at the chaff, that is the seed coats that the ants throw away, after they've opened them and eaten them, the old chaff, look, that's all small. Fresh chaff is small. In other words, they're just opening and eating these small seeds. Okay, that sounds pretty good. And in fact, if you compare, and by the way, let me see. Do I have, no, I don't have it there, but the seed size is estimated by sieving them on standard sieves. And the scale is that the smallest openings are from here and the biggest there. So the seeds increase along the x-axis. And if you look at the stored seeds, the ones from the chambers, this is from a whole bunch of different colonies, you see that they're mostly large, but the chaff is mostly small. So, okay, they got chambers full of big seeds that they seem not to be able to eat. Then to make sure, and you gotta hammer away at a question, right? You gotta anticipate what might be wrong with your thinking. So we did an experiment where we robbed foragers coming in with seeds. They're coming in from the field. And then compared that to the chaff. So the chaff, as you know, was all small, but the seeds they were bringing in were a lot bigger. So they're bringing in seeds that just pile up in the chambers. And I told you that the colonies move on average once a year, and they move all the seeds and all the contents. In fact, here's a colony in the process of moving. This is the old colony, and they've even made a highway to the new one. So they're digging the new one here. And when they move all the seeds on about the third or the fourth day. And by the end of about 5 or 6 or 8 days, it's all done. They're all in the new nest. And by the way, the new nest looks just like the old one. Now you're going to ask me, why do they move? It's the same answer I have. Why did they collect charcoal? I have no idea. But you can carry this further. During a move, again, the transported seeds are on the large size, whereas the chaff is small. So I'm just sort of emphasizing that there's a whole lot of seeds that they seem not to be able to use. So here is a seed chamber, and these usually occur in mid-depth, so 50 to 100 centimeters from about that deep to about that deep. The nest may be 2 meters or more deep. So they're in the middle range chambers. And this is a pretty typical seed chamber. There's not totally filled, but there's just enough room on top of this pile of seeds for the workers not to bump into the ceiling. So they leave enough space so they can walk around. Okay. So obviously the next question I have to answer is, what size of seeds can the ants open? Because they have all these big ones, little ones, and so on. So what can they open? So this is a standard testing sieve. So we separated the seeds into 4 different sieve sizes, and there's the dimensions, but these are the seeds. And then I marked them with a fluorescent printer's ink in 4 different colors. And this is under visible light. Each size was a different color. So here you see the big ones are blue, the littlest ones are yellow, and everything in between. So we offered these to laboratory colonies. And the ants collected all of the seeds, but they only, the measure for whether they use them or not is whether the husks appear in the trash pile in the lab. And sure enough, doesn't take long, maybe 5 days or so, before most of the small and medium seed husks are in the trash pile, but none of the large and very large ones. So in the lab, they're not able to open these large seeds. So here's another way of showing it. These are the seeds that are in the arena, and you can see very few of the small and medium ones are left in the arena, but most of the large ones are collected too. These are in the nest. There's the big ones, little ones, and the ones that are eaten, we know that because the husks are in the trash pile, are only the medium and the small. So is that an artifact of the lab? Well, we can test it the same way. Offer fluorescent marked seeds to field colonies. And sure enough, there you go. The trash pile, very soon, the trash pile contains the husks of small and medium seeds, but essentially none of the large. You can see where this is going, right? Well, you know, there's two sizes of workers. There's the big-headed guys, and they have a reputation of being seed huskers. That's the literature says that. I mean, it's one of the standards of harvester ant literature. The big workers are there to open seeds. Okay. So we had colonies with major workers and without major workers. And sure, yeah, when you had major workers, there were a lot more seed husks in the trash, but they were all the same two sizes. The big ones didn't appear more frequently. So no, that's not what they're doing. Yeah, they husk seeds, but they're just a big increased frequency, not bigger size. So then you think, okay, maybe the ants are waiting for the seeds to germinate. I already made that hypothesis. So here's some germinating seeds. This is, oh God, it's a euphorbia. I can't remember the name right now. This is a pretty big seed, so it's easier to work with. Yeah, it stays on a number 14 sieve. So we offered colonies germinating seeds of this kind and ungerminating, and we marked them with different fluorescent colors. So the non-germinating were marked with green fluorescence and the germinating with orange fluorescence. And sure enough, the husks of the germinating ones appear very quickly on the trash pile, but the non-germinating ones do not. So they're not. I mean, yeah, they can eat germinating ones. So, but then the question is, do they eat those seeds that are germinating? So we feed them dyed seeds in the lab, which is obviously easier. Germinating seeds, there's a dye called rhodamine B. It's one of my favorites, and yeah, it's kind of a fun dye. A very strong fluorescence, and you can so easily contaminate your lab. And actually, those of you that subscribe to my Substack essays, I wrote one called Contamination, because I had a student. This rhodamine B is a very fine powder, and you don't have to be careless to have it fluff all over the lab. So when she was done, you wipe a lab table and there's all these little brilliant orange red dots fluorescing under UV light. So that was miscontamination. Anyway, so we marked, I soaked these seeds in rhodamine B, and they take it up quite nicely. And then I marked the husks with orange printer's ink. Now, the problem is, how do you mark seeds that are not germinating? Okay, well, I solved it by borrowing this little tiny drill press from the physics department. And with it, I was able to drill holes 1/10 of a millimeter in diameter in the seed coats of these number 14 seeds. And then I soaked them in a blue dye, methylene blue, and then plugged the hole with printer's ink, I think. So here's the result. The flesh of these seeds is dyed blue. So now I have ungerminating seeds with blue flesh, and I have germinating seeds with orange flesh. So we feed those to the colony. There's the combination. This is under UV light. And this is the result. After 24 hours, most of the larvae are fluorescing pink, which means they've eaten the germinating seeds, and there's no blue ones. So they're not eating the blue, they're not opening those big seeds, okay? So that's pretty good. And this is under UV light. It's like little Chinese lanterns. I love this. This dye is wonderful because you can feed it to an insect, and under UV light, it looks like a party. Love it. Okay, which one? That one. So, of course, this is in the lab, and I test it in exactly the same way in the field, offering them the dyed germinating, the undyed non-germinating seeds. And after 3 days, the larvae in these now wild nests there's a lot of them are died, like you see here. They've eaten the germinating seeds. And ferrate pupae are the ones that are undergoing metamorphosis. So they're a little older, but they also have eaten the seeds. So yeah, they do it in the field too. So then the question arises, well, do the seeds actually germinate in the chambers, and under what schedule? Because obviously they're seeds of 50 different species, and they have a different regime under which they germinate: season, moisture, temperature, and so on. So this is where we did these experiments in which this is a big experiment. That's why I had Nicholas Hanley do this. He was very dependable. It had a field component in which we buried seeds of 4 different sizes and dug them up every 2 months, so 6 times a year. And in parallel, we were testing the germination of the seeds in the lab under controlled conditions. These are wine coolers, which you can buy for $56 at Home Depot. Perfect for this experiment. And this was, I think, this was even cooler. Anyway, 4 different temperatures and 4 different burial depths in the field, 4 different lab temperatures. And then we tracked which seeds of which, and these are now categorized only to seed size. We can go back to species, that's possible too. So the blue shading here is the mean temperature over the year. And then the individual lines are the germination in the field. So this is percent germinated by month. And you can see that there's very different patterns depending on the seed size, and therefore different species, right? There's number 14 and 18 seeds, which are multiple species, germinate fine in the fall and the spring, and then very depressed germination in the summer when it's hot. So look at that, okay? It's hot in the summer, they don't germinate. It's intermediate in the spring, they germinate, and so on and so forth. The ones on the blue line, number 12 seeds, are pretty insensitive to temperature. So you see that here. This is now the lab temperature from 10 degrees to 32 degrees. And you can see that the blue ones, number 12s, are kind of insensitive except at the extreme. And that's largely true of the number 20s. But the ones that take the dip have very high germination at intermediate temperatures. So every species has a set of conditions under which they tend to germinate. That's a big surprise, right? I'm sorry, I'm not a botanist, I'm an ant guy. The depth of burial didn't matter because, in fact, the temperature of the soil column up to the 80 centimeters doesn't vary that much. It's really an annual pattern, not that much depth difference. And if that doesn't blow your mind yet, this is going to be on the quiz, so you better pay attention. This is now by sieve size, 12, 14, 18, 20. I'm not expecting you to really look, but I just want you to note that there are different patterns at different temperatures for different size seeds. So in other words, each species of seeds has its own optimal temperatures and season under which they germinate. So now go back to ants. The ants are dealing with this, right? So do the seeds actually germinate in the chambers? And if they do, what happens to them? So this was an experiment I ran with Julio. We dig a pit next to a harvester ant colony and then go in from the side until we just nicked a seed chamber. So here you see a seed chamber just barely nicked. And we take this curvy piece of metal and push it through so that part of that same chamber is now isolated from the rest. The chamber has only one connection to the vertical shaft, right? So now you've got part of a chamber that the ants have access to from the central shaft, and part that they don't have access to. So what happens to the seeds? Anybody want to guess? Well, there you go. When you're done, these are, oh, it's died. It's something like batteries, probably. Oh, never mind. I got a stick here. Can't depend on batteries. All right, so obviously here then we have seeds that are germinating are all circled in red here. And up there, summary is when access is denied, there were 21 germinating seeds out of 4,400. And when it's allowed, there were 6,500 seeds and only 1.3 that were in the chambers. In other words, the ants are getting those seeds, removing those seeds as soon as they germinate. And probably, well, we know they make ant bread. So they're removing those seeds and processing them as food for their larvae. Pretty cool, huh? So the upshot is that the harvester ants do eat these very large seeds, but they do it with help from, actually, I guess it's the season, the water, the nature of the seed species themselves, and so on. So the ants are betting on a future. So let me see. Yeah, well, okay, I'll get back to this question in a second. So seed composition, as I told you once already, changed through the ages, and these are these here are the names of seeds. You guys can, I don't know, I can't even read it, so you probably can't either. Names of the more common seeds, and then these are the sizes: 20, 18, 14, 12, and you can see that that the frequency is quite variable with the seed size. These are the seeds from 1989, from the 31 colonies, and they're pictured here by the really biggest ones and then down through the smallest ones. And these are mostly grass seeds. So, and if you look across colonies now, these are 9 different colonies, and this is the seed size frequency distribution for a single colony, but all the different chambers. And you can see the chambers are all the same. They're not sorting the seeds by size. They're pretty much homogenized within the colony, but the colonies have very different seed size distribution, probably because they're in different places. And the plants that whose seeds they have access to are not the same. They differ across the landscape. So here's then the summary. These are the seeds that the ants can and cannot open by themselves. So the only ones they can open are the ones in blue here, up to number 18 civ. And then all of these pink ones, they can't open. So if you think about it, they're playing a very cool gambling game because, okay, that giant seed is 90 times as large as the smallest seeds. So, but if they can't open it, it doesn't do them any good. They would have to open and eat 90 of the small seeds to get the nutritional value, the caloric value for one big seed. So this is the gambling game that they're playing. And you know, these are all, this is the factor relative weight and relative to the smallest ones. And you can see it goes up, up, up. And then these are the species. Smart, right? Didn't I say smarter than the average ant? So this is what they do. If you look at overall, this again, the 1989 sample, 70% of the weight of the seeds that they store, they cannot open. So that is their what do you call it in gambling? That's not their stake. Bruce, you're a gambler. What's it called? Anyway, that's what is at stake for them. They have 70% of the seeds that they worked hard to go out and collect. And remember, foraging is a very dangerous profession. Once a worker becomes a forager, her life expectancy is 2 or 3 weeks. So this is what they do at the end of their life. Every seed is an investment for the colony, and every seed is a gamble for the future. Okay? What can I say? Anyway, there's a brief summary. Ants collect the seeds that are too large to open. They eat the large seeds only after these seeds germinate. They move all the seeds when they start a new nest or relocate. And because they can't open these big seeds, the big seeds accumulate over the lifetime of the colony. They become a larger and larger fraction of the seeds that they have. And these colonies, I estimated their lifespan. Once they're across the juvenile age, it's about 3 or 4 years. They have an average lifespan of 30 to 40 years. So that's a long time to be accumulating all these big seeds. They leave nothing behind when they start on average every year. When they make a new nest and move into it, everything is moved. All the seeds, they even move and the brood, of course, but they even move young workers. They carry them upside down, kind of fun. And then at the end, they move the charcoal too. Got a question? That's a good question. Do they ever get tired of waiting for seeds? How do you ask that question? Very rarely. If you feed them bird seed, that's not a seed that has any dormant period. They germinate immediately, and they often discard those. I'm not sure why, but I don't do that. I'm not mean. I don't feed them that crap. Leave it for the birds. Anyway, as I say there, number 5, ants are very patient, and it's really a species that is exploiting the seed bank in an ecosystem over a very long period of time. And clearly this has evolved over probably millions of years. Okay, that's my story. Yeah. Todd, you're in front.
If they're moving those big seeds and sort of getting more and more decades, they're low probability, but high payoff. So in a way, you're accumulating high payoff, but low probability.
Yes, Todd's point is that if the large seeds accumulate over a long span of time, how did you phrase it? It's an ever larger, the high payoff, yeah. Yeah, right. Although, yeah, I'd have to think about that low probability because that's very dependent on the temperature and moisture regime and the species they collect. And of course, bigger colonies would use a lot more seed. So you'd have to look at the accumulation proportionally. Yeah.
How many of those are wild? After all that time?
That's actually a good question. Todd is asking how many of those old seeds are viable. And I did try to germinate the 1989 seeds, but several years later, I don't remember how many years later, maybe a decade later, and germination was rather poor. On the other hand, the 2014 seeds that we collected more recently, well, you saw the germination of those. It's actually quite substantial depending on temperature and season. Yeah. So, yes. Question is, do they have charcoal only around the openings or do they ever bring it in the chambers? A. Yes, it's only around the opening. They do not bring it even into the top chambers. Or if they do, then it's very quickly removed to the disc. Now, I know you're going to ask, why did they collect charcoal? And I told you once already, I don't know, but there's 2 or 3 of the Western Pogonomyrmex species that collect and decorate their nest disc with small pebbles about the size of these charcoal bits. So that's not an answer to a question. That just removes the question by one species. Why do they do that? I have no idea. No, it doesn't. We tested that. Yeah, but then all organic matter absorbs moisture. Could be. Yeah, there are several hypotheses about charcoal that are, I agree, are very appealing, but none of them worked out. You can test them experimentally and yeah. Wilson. Oh, a good question. Wilson's asking how far do they move when they move on their annual move? An average of 4 meters is just from me to Todd. And since I tracked them for 10 years, I could track the wanderings of individual colonies. And all they're doing is wandering around it's a random walk. They're just wandering around some central point. So, yes. Does anything rot in the colony? Mm-hmm. Oh. Well, the question is, why don't things rot? And well, there's a whole ecosystem in those nests. There are colambula, which are these tiny little insect-like creatures. There's always burgeoning populations, and they probably feed on any fungus that grows on this. Then there's a spider that is found only in the nests of Pogonomyrmex badius. It's not found anywhere else. And it makes a little divot in the ceiling of the chamber, lays its eggs there, and then spins silk over it so the ants can't detect it. And that spider has a very limited dispersal. So the relatedness of spiders in a local population is often quite high because they don't disperse very far. Bruce.
Seed that they're eating, and or what effect the ants might be having on the ecosystem plants.
Yeah, Bruce asked the obvious question I couldn't answer, which is what proportion of the plant seeds are being removed from the seed bank. I really don't know, and I'm not sure how I would go about that. That would be beyond my capabilities, I think. Yes.
It's often in the literature that plants have perceived or ant dispersed, but I wonder how you might imagine that harvester ants could benefit plants dispersing their seeds, or if they cannot imagine that, could they just bring them to the nest and there they die or they get eaten?
Yeah, the question is, there are the seeds of many plants that have little attachments that cause ants to collect them, and they gnaw off the attachment and then discard the seed, then the seeds can then germinate. That's not the case with harvester ants. Remember, they're bringing them down 30 to 50 to 100 centimeters below ground, and they're waiting for them to germinate. And a seed that germinates 50 centimeters down is a dead seed, partly because ants eat it. Pardon? Right. That's right, yeah. The ants that collect those elastomes are not harvester ants. There are quite a few other species. Yes, question in the back. Yeah, Another good question. Are there seeds the ants don't collect? And again, that's like Bruce's question. I don't know because I don't know what the natural frequency of all those different sizes and species are. I just know what I collect in the chambers. David?
I got, right, that's like from the colonies is from underground, like they don't bring any of that up.
Yes, the trash is all on the surface.
Really?
Yeah, the trash, they're very sanitary, okay? They bring up all the trash and put it, usually it's along one edge of the nest of the charcoal disc. And over a period of time, as the colony excavates more and more sand, more soil, it gets covered up. So there's always the one side or the other of the nest disc, which is packed with chaff. And a lot of times that chaff is bound by mold and so on. Yeah.
Okay, yeah, maybe it does get back, because I was thinking the question off the mold, possible mold down there, is if that, one of the reasons they moved their nest is because it's been, and they, maybe not down below like I thought, but maybe up above.
Yeah, the question is, basically you're asking why do they sometimes move? Maybe it's contamination with mold and so on. Yeah, I mean, it could be. I mean, the most likely hypothesis for their annual move is sanitation. Okay, that's true. But they also have pretty interesting relationships with other microbes. So for example, in many colonies, the floors of, especially the upper chambers, are black, and it's a fabric. You can pull it up like a rug. And I'm sure it's fungus, it's fungal filaments, but it's not necessarily detrimental. It's some kind of complex relationship that they have with among other things, this particular fungus. Yeah. You know, it is literally an ecosystem underground. And I'm only just barely scratched the surface of how complex it is. My former student, Christina Kwapich, has done a lot with it. And she's extremely observant and has seen all kinds of stuff that me and my, quick and dirty have missed. Yeah. Yeah. Yeah, let me see. Go back to the, okay. This is a very large nest, but the pattern is similar. The seeds would be in that sort of middle elevation, 50 to 100 centimeters below ground. The brood and the queen are mostly in the bottom third. And by the way, those foragers, the ones that are giving their lives for the colony, right? You never find them deeper than 10 centimeters below the ground surface. In other words, they do not go deep in the nest. So the stuff they deliver is left at the loading dock, the top 2 or 3 chambers. And from there, there's another category of middle-aged ants that then distribute those things that the foragers bring in and carry them down to the consumers or the seed chambers in the nest. So it's another category of division of labor by age. Yeah. Oh, and by the way, evolutionarily speaking, the reason why foragers never enter deep in the nest, and it's not limited to this ant, it's also true in other ants, is because obviously they're out in the field and they can bring in contagion and disease. So the individuals that forage are, in evolutionary terms, not allowed back in the colony. They just bring the goods. Yeah. No, the question was, why do the tunnels spiral? No, but that's, it's a feature. I've not seen it in any other ant, but it's one of the beauties of this particular architecture. I mean, I love it, but I have a hypothesis if you want to hear it. Okay, let's say that instead of being a helix, it's a vertical shaft, right? The ants are carrying seeds. So they get into the vertical shaft and they happen to drop it. It goes all the way to the bottom, right? So if it's a helix, it can't go very far. Just a hypothesis. Yes.
So you guys, did the size sorting for the seeds that you collected from inside?
Mhm. Yeah.
Did you also look at the size distribution of the seeds as they were bringing them in? Like, do they collect more of the little seeds that they don't last as long, but they end up storing more of the big seeds?
No, I did show that slide. They're collecting mostly large seeds. Well, if you think of it as a half-life, that is the lifetime of a seed once it's collected, the small seeds would have a much shorter half-life. So you wouldn't see them accumulate very quickly. No, that's not what I meant to say. You better answer that. All my colors are unknown. Yeah, I have to think about, there's a mathematical relationship here about the rate of collection and the rate of survival that would determine, yeah. Josh?
Is it possible that as they accumulate more seeds, the benefit of giving them that seeds might be exposed to new conditions than a new fruit might?
I never thought of that. The suggestion is that maybe the benefit of moving to a new and different nest is that it provides different conditions for seed germination. Yeah, why not? You got a job. Yeah, Annie?
Their whole colony.
You would think that. Yeah, the question is, do they move because they're bigger? No, somewhat, but they're building a new nest which is very similar in size and structure of the one they just vacated. I mean, I did that, you know, I did that for a large number of colonies. And by the way, the beauty of knowing about their moving is that if I keep track of when colonies move, and by the way, they move mostly during July and August, so the summer months, about 60% of them move in those 2 months. And if I survey them, I can find the ones that have just moved, and I can make a cast of the empty nest, so I'm not killing colonies. I developed that many years ago because, you know, these are creatures. They're not just flies on horse dung, you know. They're organisms that live for 30 or 40 years and have huge territories. If I'm carrying on too much, you can stop me. But Christina, my student, showed that when you restrict, when you restrict the foraging range of foragers in a colony by fencing the nest, so the foragers can't go out in the field to collect seeds and come back. She fed them in those enclosures. Their longevity is greatly increased, so they live quite a bit longer. So being out in the field and foraging is lethal. So then she also did the opposite experiment. She fenced all the neighbors of colonies. And the focal colony that was unfenced, the forager life expectancy increased by quite a lot. So it implies that they're dying in confrontation with neighboring colonies. And a territory of a harvester ant colony can be as big as this room. So imagine a little ant 5 millimeters long, going way out to the corner there and doing battle with a neighbor and dying. I mean, if you think about it, it's just mind-boggling. Yes, Bruce.
You told us about their ingestion. What about their excreta? Do they have any?
Yes, Bruce is asking about their poop, and yes, they do. They take that out in the trash pile, as best I know. Yeah. It's at the edge of the charcoal disc. Could be. Yeah, that's true. Yeah, the antimicrobial action is being suggested, but this isn't activated carbon. They're just the butt ends of pine needles that didn't burn. So they're not really activated in the sense of absorption, chemically activated. It's just charcoal. Okay, well, I thank you for your attention. Thank you.
Thank you so much. I have to say, my favorite part is picturing you guys stealing the seeds from the little ants.
Yeah, it was mean, wasn't it? Yeah. Oh, right. Field trip, you've heard, we're going to meet on Saturday at 9 o'clock in the Lake Ella Publix parking lot, and you'll see there'll be a cluster of people there. And for those of you that either missed that or find it more convenient, we will stop briefly at the sewage treatment plant at the corner of Spring Hill and Capitol Circle Southwest. So if anybody is there by 9:30, then we'll meet you there and then continue back down to Ant Heaven. Okay, so that's the field trip. You asking about my Substack? Or? Yeah, I've been writing Substack essays now for almost 3 years. And every week, every Friday, I publish another essay. They're mostly fairly short and they deal with a range of subjects, anything I find interesting, right? And it can be ant biology, it can be swamps, it can be stories from my own life, from my youth, from whatever, anyway, just things that I find interesting. And if you want to subscribe to it, it's my name, waltertschinkel.substack.com. It's very simple, and you'll see. See? And he thinks they're good. Okay, well, thank you.